Good morning, students. Today we are going to start chapter number next plant anatomy, that is introduction and importance. After importance of plant anatomy in plant taxonomy, we must see the next point, that is importance of plant anatomy in pharmacognosy. Pharmacognosy is an applied branch of botany which deals with the biological, biochemical and economical features of natural drugs and their constituents. It is a study of drugs that originate in the plant and animal kingdom. Pharmacognosy is concerned with the history, distribution, cultivation, collection, selection, preparation, commerce, identification, evaluation, preservation and use of drugs and economic substances that affect the health of man and other animals. In these concerns of pharmacognosy, the science of anatomy has great scope and applications as mentioned below. First, the purity and quality of drugs may be evaluated by anatomical studies. Microscopical analysis gives exact idea of structural details of the drugs of plant origin. The powdered forms of drugs or drugs material in the form of certain plant parts can be identified for its chemical constituents. It is called microchemistry or chemomicroscopy. Second, lignin nature of drugs may be identified by a drop of fluorogluconol and HCl. It gives red stains with lignin. Mucilage is stained pink with rhomatium red. Cellulose swells and dissolves in coaxum. N50 iodine solution stains are starch and hemicellulose blue colored. Then next, by taking this section of plant drugs material, cell wall, trichomes, fibers, vessels, etc. may be confirmed. It helps in identification of crude drugs. Example, Noxomica has lignified trichomes, Senna drugs has vertitrichomes, while mint has glandular trichomes. Next, with the help of quantitative microscopy and linear measurement, drugs may be identified. This includes stomatal number and index, length of fibers, size of starch grains, etc. Senna varieties may be differentiated by differing stomatal number and palisade ratios. The next, cardamom varieties may be identified by number of sclerenchymatous cells per square centimeter. Cinnamon has starch grains of diameter 10 micron and such drugs can be detected correctly. Then leaf drugs may be identified by their palisade ratio. Palisade ratio is average number of palisade cells beneath each epidermal cells. Leaf powdered drugs thus can be identified. The next, stomatal number is average number of stomatal per square mm of epidermis of the length. The next, stomatal index is the percentage that the number of stomata formed to the total number of epidermal cells. Each stomata being counted as one cell, it is calculated as below. SI is equal to S/E plus S into 100 SI, stomatal index. S is equal to S for number of stomata per units, E for number of epidermis cells in same unit area. By this technique, number of leaf drugs such as belladonna, coca, datura, senna, etc. may be identified. The study of wood, bark, anatomy of rhizome, seed and flower gives idea of drugs material. It is one of the exact technique to identify such drugs from a mixture or adulterate material. Example, sandalwood has thick fibers, oil in all elements, parenchyma, cells encircles vessels in the xylem. Cassia bark has needle-like crystals in the parenchyma and abundant starch grains. Then leaf drugs of digitalis has absence of calcium oxalate crystals and oil. This indicates that there are several anatomical characters that help in identification of crude drugs and thus becomes valuable tool of evaluation of plant drugs products. Then next point, that is importance of plant anatomy in wood identification. Xylem is one of the important elements of vascular cylinder of plants. Its amount and nature varies from species to species. Thus, this feature is of great importance in identification of wood in timber industry. Xylem wood is composed of vessels, tracheids, wood fibers and a patch of xylem or wood parenchyma. These components of wood vary in their position and amount. On the basis of wood identification is possible as mentioned below. Number 1. The presence or absence of vessels and arrangement of vessels in wood is one of the important criteria for wood identification. Example, when vessels are absent, wood is described as a non-porous wood. Example, gymnosperm coniferous wood. When vessels are present, wood is porous wood type. Example, angiosperm and dicot wood. Then number 2, porous type of wood has peculiar distribution of vessels in wood and thus it may be of two types, that is diffuse porous wood and ring porous wood. So first we see that is what is meant by diffuse porous wood. So here in diffuse porous wood, vessels are more or less equal in diameter and are uniformly distributed throughout the wood. Thus, when there is a gradual changes in size and distribution of vessels throughout the growth rings, the wood is known as a diffuse porous wood. Example, acacia, that is babul, populus, that is salix, populus and salix, etc. Then second type, that is ring porous wood. When wood consists of vessels of different diameters forming different definite rings, this wood produced at the beginning of season and vessels distinctly larger in size than the late wood which is produced at the end of the season. Such wood is called as a ring porous wood. Example, Fraginus, Quercus oak, etc. The wood formed in a spring season with large sized pores or vessels is called as a spring wood or early wood. The wood formed in autumn season with a small pores is called as the autumn wood or late wood. Then third point, on the basis of secondary wall thickenings, the primitiveness or advancedness of wood can be confirmed. Thickenings may be annular, spiral, scalariform, reticulate, simple and bordered, pitted, etc. Then number 4, structure of rays in the wood is also working as a criterion for identification of wood. Example, uniseriate rays in coniferous wood, while dicot wood may be one or many cells wide. Ray height also changes from one to other species. Then number 5, parenchyma is in relation with vessels also changes and used in criterion for wood identification. Example, presence of apotracheid parenchyma speaks for primitiveness of wood, while parectial parenchyma of the advances of wood. In parenchyma is independent of vessels, while in paratracheal type, vessels are associated with parenchyma. In different types of woods, parenchyma is one of the criterion for wood identification. These varieties of criteria may be used for wood identification. Thus, plant anatomical studies have great value for timber industry also. Okay, thank you.